I was honored with the Rachmiel Levine Prize from the City of Hope. It's a career award for clinical research and mentoring. You always have to start off with disclosures, and I have been a consultant for Vertex and for a new company called Med. I serve on a data safety monitoring committee for Novartis, and I'm co-founder of a company called Stability Health, which I'll just mention. My story starts in 1984 and 1988. I had just finished my internal medicine residency at Duke and was assigned to the Navy Hospital in San Diego, which we called the Pink Palace in the Park. I thoroughly enjoyed being an internist. It was a fabulous experience. But then in 1986, I was partly responsible for inviting new lecturers for the internal medicine residents at the Navy Hospital. And I invited this man, Frank Gisari, who's a National Academy of Sciences clinician scientist at Scripps, and he studies hepatitis B. And his lecture that day was about a mouse model that he had developed to understand whether the liver failure that accompanies chronic hepatitis B is due to the virus killing the liver, the immune response attacking the virus in the liver, or a mixture of both. And as he was talking, I was so excited, I was sitting in my chair shaking. It was just so cool to me. But I looked around at my colleagues and asked him why they were falling asleep. So I asked Frank why he chose to talk about a mouse model to a bunch of physicians. And he pointed his finger at me and said, "Dave, you know as well as I do that I could give the very best therapy we know to patients with chronic hepatitis B, and I would be absolutely wasting my time because we have no idea what we're doing." And then he said, if you get what I'm talking about, pick a disease that you want to have a chance to understand why it occurs, really the fundamental basis of it. And you might waste your life too, but devote your life to that. So I've told him since he might as well have punched me in the jaw because it had that effect on me. It just really awakened me. It was an epiphany. And I looked at my schedule for the next day and it was filled with patients with diabetes. And my first reaction was, well, this will be a fun day. These are my friends. I've gotten to know them over the last couple of years. And then it dawned on me that we really don't understand the disease at a basic level. And so that night, I committed my life, that's been now, 37 years ago, to understanding diabetes sufficiently well so that we could come up with more rational treatments for it. And that became my holy grail. And then, irony of all ironies, 2 years ago, my 30-year-old son was diagnosed with type 1 diabetes. So now it's not just a professional passion, it's got a personal characteristic to me as well. So this is the Levine Award, and so I went back and read one of Dr. Levine's papers, and in 1967, he wrote a review in New England Journal that said the biography of insulin is constantly being amended and rewritten. Some parts of the story will persist longer than others, but the more ephemeral portions serve an excellent purpose too. They lead to work that results in a more permanent record. So I'm going to tell you some fits and starts of my career and how I think that the field is moving forward, but always some surprises. So obviously there's lots of people to thank over a 37-year career, and I'll mention many of these people as I show the work I did with them, and I'll highlight certain individuals in particular. But I have to thank also that a lot of stuff that we've done through the years has been because family members on a worst day of their life, when they're told that a loved one is brain dead, has agreed to share their loved one's organs so that we can study it. And so we're very thankful for the generous contributions of the donors and their families. So let me say a word about Rachmiel Levine. Most of you probably don't know who he is or what he did and why the Levine-Riggs Symposium is my favorite diabetes meeting of the year. But Rachmiel Levine was born 113 years ago in Poland. His mother and father died when he was a young man. He discovered some American relatives, but he couldn't get a visa, so he immigrated to Canada and was adopted there by a Canadian physician. And in the 30s, he decided he wanted to pursue a career in medicine. And prior to his work in diabetes, the conventional thinking was we all take these things for granted nowadays, but the concept of a cell membrane that was impermeable to larger solutes was not well appreciated back then. And the conventional thinking about how insulin worked was that glucose in the bloodstream passively entered the cell, and then insulin somehow stimulated the metabolism of glucose inside the cell. And what Levine proposed was that what insulin really did was let glucose into the cell. It didn't just passively flow. Insulin had to unlock the gate that would let insulin into the cell. And he demonstrated that in this paper, in which he eviscerated dogs and took out their kidneys, and then infused galactose, which is a 6-carbon sugar, just like glucose, but it can't be metabolized intracellularly. And if you infuse galactose, it'll achieve a steady state in the bloodstream of these dogs, unless you give insulin, allowing it into the cells, and you can see the galactose levels fall. So it proved the transport theory as opposed to the stimulating intracellular enzymes of insulin. Anyway, my career started after I finished in the Navy out in San Diego. I went back to Duke in 1988 through 1991. I went from being a decent clinician to a terrible scientist overnight. But fortunately, our mentor, Perry Blackshear, stuck with me, and I learned molecular biology over a 3-year period in the Howard Hughes lab there. And then I went back to the Navy in Bethesda, Maryland, where I was determined to study type 1 diabetes pathophysiology. And right around that time, Kevin Lafferty, shown here, who became a friend, popularized what was called the 2-signal model of T-cell activation, which was really borrowed from an earlier model of B-cell activation, which posited that here's your T lymphocyte, and APC stands for antigen presenting cell. T lymphocytes are known to recognize antigens presented by major histocompatibility complex proteins, the antigen shown as this triangle. And prior to Kevin coming up with the 2-signal model of T-cell activation, it was thought that this alone was enough to activate the T-cell. And what he said is that can't be enough. And he proposed that if a T cell saw its antigen but didn't, and that's all that happened, not only was the T cell not activated, it would be permanently inactivated. What he said you needed to activate a T cell was this specific recognition event triggered through the T cell receptor, but also a second signal, which he called signal X or signal 2 at the time. And another corollary is that if a cell got signal 2 but didn't have the recognition, nothing would happen. Well, it turns out that the first receptor ligand pair that appeared to confer this signal 2 function was a receptor ligand pair called CD28 on the T cell and CD80, or it was then called B7 on the antigen presenting cell. And that biochemical function was first reported by my mentor in the Navy, Carl June, who's now famous for developing CAR T cell therapy for cancer. Anyway, Carl and I agreed that he would figure out how to turn on the immune response to cure cancer, and I was going to figure out how to turn off the immune response to cure diseases like type 1 diabetes. Now, that simple cartoon was recently reviewed by Jeff Bluestone and Mark Anderson and published in New England Journal a couple years ago. And here again, you see the T cell with the CD28 co-stimulatory receptor, which will be triggered by either CD80 or CD86 on the antigen presenting cell. And So my hypothesis, that was a brand new theory in the late 1980s and the early 1990s. And so I proposed a hypothesis that maybe type 1 diabetes, the immune destruction of pancreatic beta cells, was caused because the pancreatic beta cells were making CD80 or CD86, and that was enough to trigger a T cell response against the beta cell. That was the hypothesis. Now, to test that hypothesis, you would want to make a transgenic mouse that would express constitutively. CD80 was the first one I made on the pancreatic beta cell. And lo and behold, that year, Gordon Freeman at Harvard had cloned human CD80, and I called him and he was willing to share the clone with me. And Don Steiner, a giant in diabetes research at the University of Chicago, this was at the early days of PCR. And so I could have PCR'd their promoter, but Don Steiner sent me a plasmid with the rat insulin promoter. And I made a rat insulin promoter mouse driving CD80 on the pancreatic beta cells. And lo and behold, those mice, and I'll never forget it when the first mouse turned diabetic, but because I'd spent 3 years on this work. But those mice were highly susceptible to immune-mediated diabetes. So we published this stuff in good journals, PNAS, Diabetologia, Diabetes, in various ways, shapes, or form. But what we're really after is the truth. It's true that you can make a mouse that is highly susceptible to anti-beta cell autoimmunity, but is that what's happening in people? And in the mid-1990s, a group in Japan, if somebody was diagnosed with type 1 diabetes, they would actually do a endoscopic pancreatic biopsy. So I contacted them. It was Professor Hanafusa and Akihisa Imagawa, and asked them if I could get some sections of a human pancreas biopsied shortly after type 1 diabetes onset. And the bottom line is we looked at 16 different patients with recent onset diabetes, and we saw no evidence that the beta cells were expressing CD80 or CD86. So I call this the great tragedy of science, the slaying of a beautiful hypothesis by an ugly fact. So, I'm after the true cause and treatment for type 1 diabetes. This mouse model was really cool, but it wasn't the true cause of diabetes. So what do we do? And I have to credit Adam Edelstein with helping me with the graphics here. So Adam, if you're on the call, thank you. But here was my holy grail. And all of a sudden that door slammed shut. But I thought, okay, now what am I going to do? It required a change in direction. So what we knew from this rat insulin promoter CD80 transgenic mouse is that the mouse was very susceptible to autoimmune beta cell killing, and we could control the timing of the killing and we could control the pace of the killing. So I thought, even though this was not what causes human type 1 diabetes, maybe we could learn something about the beta cell response to an ongoing autoimmune process. And it was a mouse, so you can euthanize the mice whenever you'd like. And one of the things that we found is that you can take islets from a mouse and break them into single cells and then sort them on something called a cell sorter for size and granularity. Granularity is just a measure of how many insulin granules are in those beta cells. And what we found is that as mice are developing autoimmune diabetes, their beta cells get larger. You can see there are more of them over here than here, and they lose their granularity. They're losing their insulin granules. I can't swear that that's what's happening in people because there's no way to know. We don't study identical twins, one who doesn't have diabetes and one who does, and get their pancreases out. But my hunch is that this biology exists in people too, that the beta cells are stressed, they grow large, and they degranulate. The other thing that we started doing is the traditional way up until about 20 years ago to count the number of beta cells, alpha cells, and delta cells in an islet was to do a pancreas section and then to sit under a microscope for hours counting how many beta cells, how many alpha cells, and how many delta cells there are. And if you want to get a severe headache and put yourself to sleep, you would do that all day, but that's what we used to do. And we reasoned that maybe we could do that much simpler, faster, and more accurately by breaking these islets into single cells and running them on a flow cytometer. But we had to demonstrate that. So we published that when you compared the cell constituents of an islet using the traditional way of immunohistology versus the simpler, faster, more accurate way of flow cytometry, you get the same answer. And what I want you to note here is that mouse islets are about 3/4 beta cells, about 20% alpha cells, and about 5% delta cells. So that's we learned something about the islets as diabetes is developing. And then we started doing similar studies in human islets. And as opposed to mouse islets, where you see the beta cells are in the middle and they're surrounded by alpha and delta cells, human cells are all mixed up. The beta cells, alpha cells, and delta cells form a gemisch. Moreover, you see that a normal human islet, and from different donors, can have as few as 22% beta cells and as many as 75% beta cells. So human islets are highly variable donor to donor. And I must say that you'll notice this is published in the Journal of Histochemistry and Cytochemistry, not a very commonly quoted journal. And it's only because we submitted it to 7 different journals and they all said, oh, well, you're crazy. Islets are 75% beta cells. You know, we've done it a million times in mice. No one believed us that human islets were different at the time. So that's another lesson we learned, that human islets are extraordinarily variable and we don't understand the biology underlying that variability. So now getting back to this review from Mark Anderson and Jeff Bluestone, we've been talking about the fact that I made a transgenic mouse that would express these things on the pancreatic beta cells. But we also knew that there are ways to inactivate the immune response by other receptors that come up on the T cell that interact with the same CD80 on the antigen presenting cell. And more relevant to what I want to talk about today is the whole field of T cell co-stimulation has grown much more complex over the last 25 years, 30 years. I've mentioned CD80 interacting with CD28 as the first co-stimulatory receptor ligand pair, but now all these other ones have been characterized. In fact, Jimmy Carter is still alive today, as you know, because if you interfere with PD-L1 and PD-1, you can turn on a T cell response, and that was his immunotherapy for his metastatic melanoma in his brain. But we became interested in another co-stimulatory receptor ligand pair called CD40 on the antigen presenting cell and CD40 ligand or CD154 on the T cell. So I'll talk about that next. And here I have to acknowledge several people. One is Chris Larson, who demonstrated that he could transplant the skin of a black mouse to a white mouse and give a blocking agent for this receptor pair and this receptor pair, and he could make the skin graft stay on a mouse, which was unheralded. You can transplant various organs in a mouse, but skin is the toughest thing to transplant. And he reported that he was having success with this pathway. So I recruited Alan Kirk, who was then completing his transplant fellowship at the University of Wisconsin. I had already been working with Dale Greiner here at UMass in various ways. And I also collaborated with Norma Kenyon and Camila Ricordi at University of Miami because they were experts in primate islet isolation. And we did this experiment. We took a monkey and cut out its pancreas. So obviously that monkey is going to have terrible insulinopenic diabetes. We took a different monkey, took its pancreas and isolated the islets from this monkey's pancreas, completely mismatched, and transplant the islets from this monkey to this monkey. And then we gave anti-CD154, 6 doses over the first month. and the animals had completely normal blood sugars. We also did kidney transplants, completely mismatched monkeys. And we had monkeys go 5 years without any rejection of the organ. And these papers were widely reported with much fanfare in all the best journals, as you can see, Nature, PNAS, Diabetes. And in fact, it got great national attention. The National Institutes of Allergy, Infectious Disease, Immunology, Allergy and Infectious Diseases reported or started a whole new network called the Tolerance Network based upon the research that we did at the Navy Medical Research Institute. And in the periodical announcing this new Tolerance Network, they cited our work. And it even made national news, as I'll show you.
CBS News headquarters in New York. John Roberts explains how this works.
This is a non-toxic, short course therapy, which for long periods of time prevents the rejection of a transplanted organ. And that has not ever been done previous to this report.
If this ultimately works in humans, it will, I believe, totally revolutionize the field of transplantation.
So you can see it was big news. Front story of the New York Times, front story of the Washington Post. I mean, it was big news. So we were invited, the whole Navy lab was invited across the street to the NIH, where we could test this therapy in human clinical transplant trials. And it all came crashing down a few years later when 10 of the 100 subjects that were treated with anti-CD154 for various things, multiple sclerosis, lupus, immune thrombocytopenic purpura, kidney failure requiring a transplant, 10% of the patients had thromboembolic complications. There was an MI, a TIA, a stroke, and one fatality of a patient dying of a pulmonary embolus. So when that happened, we had 22 primates here in Boston and at Cambridge at Biogen, who created the anti-CD154. And those animals were being treated with very high doses of the anti-CD154, and they were euthanized. And even though the animals looked fine, we found evidence of pulmonary emboli in 15 of the 22. So it all came crashing down. And there are still people at Biogen and other companies working to develop other anti-CD154 agents not associated with the thromboembolic complications. But again, another door slammed shut. And so now I was left wondering what I was going to do next. Now we tried to create our own anti-CD154. I won't tell you about that. But right around that time, James Shapiro, following on some work that David Sharp had done 15 years earlier, reported in a very widely popularized paper in New England Journal that he had cured 7 patients with type 1 diabetes by doing an islet transplant. So I was at the NIH and we had unlimited resources and we set up an islet isolation facility, and we became the first center in the world to try to repeat what the Edmonton group was reporting out of Alberta, Canada. And when I say islet transplant, what Camilo Ricordi taught me to do was to isolate islets from a brain dead donor's pancreas. And then the recipient would be taken to the interventional radiology suite, where the interventional radiologist would place a cannula in the recipient's portal vein, and I would simply push on the end of a syringe containing the islets to go into the portal vein, where they would go out to the distal regions of the portal circulation, lodge, sense glucose, and make insulin. And what did we find? Well, as opposed to what James Shapiro had done, they gave islets from as many as 4 donors to their recipients. We said, no, we're going to limit the islets to 2 donors. And of our 6 recipients, 4 achieved at least temporary insulin independence, and half of them were insulin independent for a year. But we stopped at that point, and the reason we stopped is that we saw some complications, one with a partial portal vein thrombosis. One patient had an intra-abdominal bleed. But the main reason we stopped is that the immunosuppressive agents we had to give to prevent these islets from being rejected was worsening kidney function. So our concerns at the time were immunosuppressive agent toxicity, the renal dysfunction, infections, malignancies, and other problems. The drugs included FK506 and rapamycin. A lot of patients developed mouth ulcers. We were concerned that we were sensitizing the human donors against the donor tissue. We had seen these procedure-related complications, and we knew that this was never going to be the cure because of the limited islet supply. Well, we were ridiculed when we published this negative report saying we just didn't know what we were doing. And now 13 years later, the international consortium of looking at how islet transplant did in all of these expert centers was no different than we reported 15 years earlier, 50% insulin independence. In fact, they even defined insulin independence as near normal glycemia, not true normal glycemia. And they also reported a decrement in kidney function, and that 2 donors developed donor-specific antibodies. And so basically, this large multi-billion-dollar study simply corroborated what we had said more than 15 years earlier. And WC Fields says that if at first you don't succeed, try again, then quit. No sense being a damn fool about it. So then what did we do? Well, so by the way, here's the initial excitement when Edmonton reported their results. Then our report came out and the number of islet transplants fell. Then this international consortium was funded. And with the results of it, you can see that very few islet transplants are being done now, only because it's not the cure. We were also told that the renal insufficiency problem that we had seen was because we didn't know how to properly administer immunosuppressive agents. So we did this study. We looked at pancreas transplantation, and those are done by transplant surgeons and transplant teams. And we looked at all patients in America listed for a pancreas transplant from 1995 to 2000. And those patients were divided into the 3 general categories of when a pancreas transplant is done. You can get a pancreas transplant alone if you have normal kidney function and you just want a pancreas transplant. You can get a pancreas after a kidney transplant if you had diabetes and kidney failure and you got a kidney transplant and now you're on immunosuppressive agents anyway, you might as well get a pancreas transplanted. Or you can get a simultaneous pancreas kidney transplant if you have type 1 diabetes and kidney failure, and you can get a pancreas and a kidney from the same donor. So for this analysis, I want you to imagine 2 people with identical type 1 diabetes diagnosed on the same day. And this person gets listed for a pancreas transplant, and they have to wait a period of time, and then they get transplanted. And we followed this patient for 4 years after their transplant or until they died. Their running mate, also listed for a pancreas transplant, never got transplanted. So what we wanted to know is, did getting the transplant help these, as statistically similar patients? Did the transplant improve survival for the one that got the transplant? And what we found is that if you have diabetes and kidney failure, and you look at the first 90 days after the transplant, you're more likely to die if you get the transplant than if you don't. But if you get beyond the post-operative complications, your survival is improved if you got the transplant. Unfortunately, if you have normal kidney function, and you get a pancreas transplant alone, that's either a pancreas after a kidney transplant or a pancreas transplant alone, your mortality is never better. You're always worse off having gotten the pancreas transplant than had you not gotten the pancreas transplant. And we wondered why that is. And if you look at patients with type 1 diabetes and kidney failure, and this is an important point for you all to keep in mind, there's about a 10% per year mortality for kidney failure and diabetes. But if you get just a kidney transplant, you still have diabetes. The survival improves markedly. So now you have about a 2% per year mortality, or 10% over 4 years. But if you never had kidney failure and you have type 1 diabetes, the reason why your prognosis for survival isn't improving is because it's already so good. There's only a 1% or less mortality associated with even long-standing severe and brittle type 1 diabetes. So we published that, and at the time that we published that, which was in 2003, this was the number of simultaneous pancreas kidney transplants done every year. And you can see that the growth in the pancreas transplant field was patients getting pancreas transplants alone. And we reported that, that's associated with increased mortality. And as you can see, the number of pancreas transplants has fallen every year since. Now, we've done an analysis that suggests if we hadn't published this paper and the number of solitary pancreas transplants had continued and become equivalent to simultaneous pancreas kidney transplants, that we estimate that over this 15, 20-year period, at least 100 to 150 people would have died. So this is an example of a paper that we published that we think saved lives, just like vaccines do, but it flies under the radar screen. In any case, we weren't going to do pancreas transplants. We weren't going to do islet transplants. So now what do we do? Well, when we were evaluating patients for our islet transplant protocol at NIH, we noticed that many patients, and this is now common knowledge, but it was apocryphal at the time, many patients with long-standing type 1 diabetes still had measurable, albeit very small, amounts of endogenous pancreatic insulin production. And so we reasoned that maybe the pancreas is always making new beta cells, just dripping, but that we never can achieve enough beta cell mass in the pancreas for therapeutic benefit because the autoimmune response is killing them as soon as it's making them, and that maybe metabolic abnormalities are also killing the beta cells. So that was our hypothesis. And we reasoned maybe we can weaken the autoimmune response and maybe we can control the metabolic abnormalities. And if we do that and give it time, maybe we can begin to see a person with type 1 diabetes starting to make more insulin again. And we looked at that in a couple different ways. One is, I told you that we had transplanted pancreatic islets into the liver of a patient with type 1 diabetes. And we realized that if you stimulated insulin release and measured where that insulin first appeared, if it was coming from the pancreas, you would see that insulin in the portal vein first. If it was coming from the islets that you transplanted into the liver, that insulin would appear in the hepatic vein first. So we looked at our patients that had received an islet transplant and in patients who had received a whole organ pancreas transplant. And when you do a whole organ pancreas transplant, you place the organ in the pelvis and it drains into an iliac vein. So what we did was we put a cannula in a central vein, the jugular vein, and in the portal vein, and then we stimulated insulin release. If the first place you saw insulin was here in the portal vein, it meant that that insulin was made in the native pancreas. If the first place you saw it was here, it just would mean that the pancreas made insulin was going to the heart, going through the body, and then finally getting back to the jugular vein. And we put cannulas in these veins and we measured the kinetics of insulin release. And what we found, and this is just one example of a lady who had, she was insulin independent 2.5 years after receiving an islet transplant. This was the C-peptide release measured in her peripheral vein. And this was the C-peptide release measured just downstream of her native pancreas, not the islets that we transplanted. So it would suggest that this amount of insulin appearing in the first few minutes after we gave arginine was coming from her native pancreas. 40 years after she was diagnosed with type 1 diabetes. So we did 7 subjects with a beta cell transplant, and we found that all of them had evidence of pancreatic insulin production, but it was always very tiny. And that is despite the fact that they'd had a transplant in many cases many years earlier, and despite controlling their blood sugars and despite controlling the immune response. So then we reasoned, well, maybe we're not seeing growth of the pancreatic beta cells and improved pancreatic insulin production because we waited too long. These were people that had type 1 diabetes for 10 to 15 years, and whatever regenerative capacity was gone. So we did another trial looking at patients who'd had diabetes for just 5 years, between 5 and 10 years. And they were clearly type 1. They were insulin dependent. They had anti-islet antibodies. They were lean, but they had some C-peptide, considerable C-peptide really in this peripheral circulation. And we immunosuppressed some. And at the time, there were reports that GLP-1 receptor agonists were causing beta cell proliferation in vitro. So we treated these patients for 6 months with Byetta, exenatide, or not, and half the patients in each group also got immunosuppression. And then we crossed over the patients so that those that didn't get exenatide for the second 6 months got exenatide. The bottom line is that we found that the pancreatic insulin production measurable in individuals with longstanding type 1 diabetes was a certain level at baseline, and if you gave them a mixed meal, they made more. If you very tightly controlled their sugars, the amount of pancreatic insulin production went down because we were giving more insulin, and that was during our run-in period. But then even giving exenatide for up to a year, we never saw any improvement in the amount of insulin production coming out of a human pancreas. And we also did T-cell assays because we were worried that maybe the exenatide was causing beta cells to grow, but the immune system was killing them just as soon as they were being made. But our immune assays didn't find any evidence that we had further stimulated the immune response. So we concluded that many individuals with type 1 diabetes continue to make insulin even decades after their diagnosis, that insulin production responds to physiological stimuli, meaning if you give a meal, the pancreas makes more. If you give insulin, it makes less. But we weren't able to find anything that caused the pancreatic insulin beta cells to grow. So we wanted to look one other way and just try to measure the proliferative capacity of the human beta cell. And around that time, another closed door, open window, Christy Spalding from Sweden took advantage of what is called the bomb curve. And that is the fact that we've known for the last 70 years very precisely how much C14 is in the atmosphere. And it was very low until the late 50s and early 60s when it suddenly spiked. And that's because the US and Soviet military people were blowing up bombs in the atmosphere until Linus Pauling put a stop to it. And when the nuclear atmospheric testing stopped, the amount of C14 in the atmosphere started to fall. And it's not because the C14 is decaying, because the half-life of C14 is nearly 6,000 years. This decline in atmospheric C14 is due to the fact that the CO2 is dissolving into the oceans and becoming part of all growing things. When cells divide, they require carbon, and that carbon is being taken up by living things and dissolving in the oceans. So Christy Spalding realized that this was like a pulse chase experiment for those of you who learned about those things in college. And what she did was cut down pine trees and isolate the DNA from the different rings of a pine tree, knowing that this DNA in this pine tree ring was made in 1950. The DNA in this pine tree ring was made in 1970. And she measured the amount of C14 present in the DNA DNA made of the different eras. And she found out that that correlated very well with the bomb curve. She could take pine tree DNA and tell you what year it was made by how much C14 was in the DNA. So a fellow working with me at the time, Shira Pearl, said, why don't we do that with human beta cells? So we isolated human islets from a woman who died at age 80 of head trauma with a normal hemoglobin A1C, and we measured how much C14 was present in her beta cells. And we found that her beta cells dated back to around 1950 to 1955. In other words, there hasn't been this low level of C14 in our atmosphere since 1950 or 1955. So she spent the last 60 years of her life not making new beta cells, living the last 60 years of her life with beta cells she made when she was 25 to 30. And we did that with another donor and got the same result, suggesting that beta cells can be made up until young adulthood and then not thereafter. And finally, we looked at another way too, because in the late 80s, early 90s, patients with bad cancers were treated with thymidine analogs for two reasons. One is you could measure a cancer's proliferative rate by seeing how much thymidine analog was taken up in their cancer. And it was also thought to be radiosensitizing. So if you treated patients with this and then gave them radiation therapy, maybe the cancers would die more than the surrounding tissues. In any case, we found 18 patients who had received thymidine analogs and then had their autopsies done at the NIH, and we were able to locate their pancreases to ask the question, did any of their beta cells take up thymidine analogs, suggesting that the beta cells had divided. And we found two donors who had had bad cancers, age 18 and 20, who lived a short period of time after they got the thymidine analog. And we were able to find beta cells that stained with thymidine analog. But whenever we looked at any of the donors over age 30, we never saw a single beta cell stain with the thymidine analog. So using two very different techniques, and now there have been a number of other techniques that suggest that the human can make, under most circumstances, I'm not saying it's impossible, can make beta cells up until the age of 25 or 30, and then you spend the rest of your life with the beta cells that you made up until you were age 25 or 30. So that was interesting. So we weren't going to cause new beta cells to grow. What do we do next? Well, I mentioned this is an award for mentoring, and I'm very proud of some of the scientists I've mentored. Scott Solomon-Poor now runs the JDRF Center at the University of Michigan. Jason Gallia is a JDRF investigator at the Joslin Clinic. And Annette Shalev is an investigator. She runs the Diabetes Center at the University of Alabama, Birmingham. And she did this study with me at the NIH 21 years ago. We took isolated human islets. She treated them with low glucose or high glucose, and then isolated the RNA from these two different conditions and looked to see what genes were expressed. And the gene that was most upregulated by high glucose, she came to me and says, it's a gene called TXNP. And I said, what's TXNP? I had no idea. But when she left the NIH, initially to the University of Wisconsin and now at UAB, she continued studying this TXNP protein, and completely independent of me. I want to point out this first paper I was the senior author on, but everything she's done on that since is due to her. She found that TXNP plays a role in islet health and function. She found drugs for preventing TXNP expression, including the antihypertensive drug Verapamil, showed efficacy in animal models of type 1 and type 2 diabetes. And 2 weeks ago in JAMA, there's a paper showing in a multi-center large trial that Verapamil is very effective at preserving beta cell mass in humans with recent onset type 1 diabetes, and I'll talk about that in a moment. We've continued studying human islets. We can take them and separate them into individual cells and then measure the messages made in those cells. And when we do that, and we look at donors with type 1 diabetes compared to non-diabetic donors at the genes that are upregulated, here's our old friend TXNP that turns up in the beta cells of donors with type 1 diabetes. But we also found the MHC molecules, including MHC class 2 molecules, are upregulated in the beta cells of someone with type 1 diabetes. And I won't tell you more about that. So now I want to conclude with the latest things that we're doing. And I have to give special thanks to Doug Melton, who put together this group, the JDRF Center of Excellence in New England. John Cammett, who some of you have known because he's been a member of our visiting advisory committee, and Esther Latres, who is the chief scientific officer for the JDRF that interacts with us for these studies. Anyway, Doug put together this, I consider it an all-star team of investigators, and I don't show names on here for a reason. We've all sworn that it won't be about us, that we're committed to trying to find a curative therapy for diabetes. And the minute we start focusing on us, we lose sight of the fact that this is an anonymous group effort, but they're all superstars. Judith Agudo from Dana-Farber, Mike Brem here, Callie Fu, who we just recruited, Jason Gallia, who I mentioned, you all know Dale, Sally Kent, great immunologist here, Sam Reddick, Renee Marr, Ramona Pop, Lenny Schultz, Jennifer Wang, Pong Yi, and Lynn Carr. So 13 years ago, I was asked to write a review on the status of pancreatic islet transplantation. And as I told you, one of the major limitations is we just don't have enough islets. And at the time, we dreamed of the ability of being able to grow islets. We're not going to get them from brain dead donors. It's just never going to be feasible. We dreamed about being able to make SC islets, stem cell derived islets, from induced pluripotent stem cells or from embryonic stem cells, but it was a dream until this report from last summer at last summer's ADA. And these are Vertex created stem cell derived islets transplanted into a patient with type 1 diabetes. This gentleman had diabetes for several decades. I think that we could do better with his glycemia control, but this was his glycemia control prior to him getting the SC islet transplant. You can see his A1C was 8.6%. He only had a 40% time in range, and he was getting 34 units of insulin a day. And then he got islets made from stem cells. And within 4 months, he was on 2.6 units of insulin a day with an 81% time in range and a hemoglobin A1C of 6.7. And now, 8 months after receiving those islets, he's off insulin with a completely normal glycemia control. This experiment changes everything because it's no longer a theoretical possibility, it's doable. So I call it a talking dog experiment. You can say, okay, well, you did it once, but it's only one time. It doesn't matter, it's only one time. It's proven that you can make a stem cell derived islet and it can get someone off insulin. The trouble is that that gentleman is having all sorts of immunosuppressive agent complications, just like the ones that we saw at the NIH 20 years ago. So what we'd like to do is take those cells that are growing in the lab and genetically manipulate them so that they can be transplanted without the need for immunosuppression. Well, how do you do that? And you can't give all the various genetically modified cells to people, so we rely on mice that Mike Brem and Dale Greiner have been characterizing with Lenny Schultz for the last 25 years. The idea would be to take human SC islets, put them into mice without any immune system, and then transfer into that mouse the human immune system and see if these islets can resist the human immune system. And these are studies that we're doing now. There's pros and cons of it, which I could talk about if you're interested, but I won't now. And then, of course, we have various flavors of mice that Lenny Schultz, who's a mouse genetics magician at the Jackson Lab, creates for us. He can prevent graft versus host disease by making modified mice. We can improve the innate immune system when we transplant human cells into the mice with various tricks that he employs. And we can create mice whose native beta cells die, either because they have a mutation in their insulin gene or because they're transgenic and express the diphtheria toxin on their beta cells. And you give those mice diphtheria toxin and they lose all their beta cells. And we're also working on assays where we can take single human islets and put them in a well and measure the insulin released from this single islet, because then we can also put human immune cells in there and see if we can recapitulate the immune response in a test tube as a faster, cheaper readout of the genetically modified SC islets. Thomas Watson, the former CEO of IBM, said, if you want to succeed, double your failure rate. We're looking for very rapid assays to tell us whether the SC islets will survive when faced with the immune response. And last, I mentioned that I co-founded a company now 5 years ago called Stability Health. I'll just say that we get to know the patients and their providers and do all the things that you'd ideally want to promote your patients without you having to reach out all the time. It's like the coaching program, but for everybody with diabetes. So we establish a relationship with the patient and their physician. We have electronic tools, and we keep ongoing engagement with the patient and the physician. And we're launching at several centers now. Lincoln said, public opinion on any subject always has a central idea from which all its minor ideas radiate. And I contend that our role as physicians will be to exercise our art solely for the cure of patients, recognizing that if you only focus on this, you'll go broke. So you have to do it in a way that doesn't lose money, but should never be driven this way. It should always be driven this way, in my view. So my last slide is I had the privilege of visiting Banting's home, where he came up with the idea of purifying insulin that revolutionized diabetes care. Is just off to the side here, and just outside that home is the Flame of Hope. And Banting said you must begin with an ideal and end with an ideal. And the ideal for me is a healthcare system that lives up to that name, of truly promoting health as opposed to responding to crises, and a legitimate cure for all with type 1 diabetes. And that I contend should be all of our holy grails.